Buenos días para todos. Bom dia para todos. Temos também umas pessoas aqui de fala portuguesa. Ana Maria Sechi, quero te agradecer pelas gestões para esta apresentação. Bom, Ana Maria, posso falar em espanhol também, certo? Bom, então vamos começar nossa conferência, nossa atividade do dia de hoje. Vamos a começar nossa atividade do dia de hoje. Então, primero, pues darle la bienvenida a todos los presentes, agradecer por su participación en el seminario del Instituto de Biotecnología, especialmente al doctor Rafael Ferraz Alves, que será el conferencista del día de hoy. Antes de iniciar la conferencia, por favor, ¿me confirma alguien que me escuchan bien? ¿Ivonne?
Sí, se escucha bien.
Gracias, Ivonne. Bueno, entonces comienzo con algunos anuncios parroquiales. La invitación a la celebración de los 30 años de la maestría Interfacultades en Microbiología, que está organizada por el profesor Daniel Uribe como coordinador del programa, los días 26, 27 de mayo, de manera virtual. Habrá 3 conferencias magistrales en las áreas de salud, de agrícola, de bioprocesos, y también tendremos conferencias de trabajos destacados de los egresados de la maestría. Los estudiantes, los egresados que deseen participar pueden postular sus trabajos, sus proyectos para presentaciones orales o en póster. Y se tendrá una premiación que será presencial el día 27 de mayo a partir de las 2 p.m. Entonces, los interesados de participar presencialmente pues deben hacer la inscripción porque los cupos son bastante limitados. También hay un concurso de fotografía y los ganadores saldrán en la portada y en las páginas de la revista colombiana de biotecnología. Entonces, más detalles sobre el evento los pueden tener en la página que les va a compartir Ivonne por el chat aquí de la conferencia, o la envío yo, no sé si Ivonne lo está mandando. Ah, ya está por aquí. No, todavía no. Ahí va el link para la información de toda la parte de la celebración de los 30 años. Entonces también tenemos el curso teórico práctico de PCR en tiempo real. Listo, retos y aplicaciones. Este es directamente del IBUN en los meses de mayo y junio. Listo, vamos a hablar de los fundamentos, las aplicaciones, el diseño y sobre todo la parte práctica. Listo, para tener resultados de calidad. Diseño de primers, los equipos, la cuantificación. Entonces es bastante completo el curso, están todos muy cordialmente invitados. Lógico, es un curso que tiene un costo, pero creo que el costo-beneficio está completamente justificado por ser una temática tan importante en nuestra área de trabajo. También los invitamos a inscribirse en el canal institucional de YouTube, Instituto de Biotecnología IBUN, es fácil de encontrarlo. También alguna dificultad en ubicarnos en Facebook, pues tienen el apoyo de la dirección del instituto para eso. Bueno, entonces para iniciar la presentación, les cuento, tenemos al doctor Rafael Alves de Brasil, ¿cierto? Con una amplia experiencia académica y profesional de la aplicación de la biotecnología. La presentación será de unos 45, 50 minutos y también tendremos el tiempo para las preguntas. Sugerimos que hacer todas las preguntas al final, entonces pueden irlas enviando por el chat, pueden ir tomando notas y al final de la presentación, para facilitar la dinámica, Rafael responderá las preguntas pertinentes. ¿Listo? Bueno, entonces el doctor Rafael Ferraz, ya lo tenemos aquí conectado, están viendo ustedes la foto del doctor Rafael. Es biólogo de la Universidad Estadual Paulista, UNESPI, en el 2008. Tiene magíster en Ingeniería Química de la Universidad de São Paulo en el 2013 y doctorado en Biocombustibles y Bioenergía en el 2020 en la Universidad de Campinas, ¿sí? Tres de las universidades más importantes de Brasil. Hizo una pasantía doctoral durante sus estudios en la Universidad de Minho, en Portugal, y después de eso se ha desempeñado como especialista en aplicaciones en la empresa ProSim Biosolutions, que se enfoca en el desarrollo de productos enzimáticos para diferentes áreas como biocombustibles, alimentos, bebidas. Desde el 2019 trabaja como especialista en Novozymes. Ahorita estábamos hablando sobre eso y él les va a contar un poquito sobre la empresa. Tiene experiencia en fermentación industrial, bacterias y levaduras, microbiología industrial y biología molecular. La conferencia que nos va a presentar Rafael es sobre la evolución en laboratorio, un tema muy interesante, ¿sí? Con algunas aplicaciones que él ha desarrollado. Entonces, no quiero quitarle más la palabra a Rafael para que aprovechemos al máximo el tiempo de nuestro seminario de hoy. Rafael, tienes la palabra entonces.
Sim, muito obrigado. Muito obrigado, Carlos. Gostaria de agradecer a todos pela presença e pelo convite. Fico muito feliz de poder compartilhar um pouco dos conhecimentos que foram gerados no meu trabalho de doutorado. Espero que traga bastante informação, conhecimento, né? Compartilhar o conhecimento com vocês. Eu vou fazer a apresentação em inglês, como eu combinei com o Carlos. E só, antes de começar, eu queria falar um pouco da Novozymes, que eu acho que talvez não seja todas as pessoas que conheçam a empresa Novozymes, que é a empresa que eu atuo atualmente. A Novozymes, ela é uma empresa dinamarquesa, multinacional, é a maior produtora de enzimas do mundo, é um grande player na área de enzimas industriais. Aqui ela está espalhada em vários lugares do mundo, então a sede é na Dinamarca, mas temos no Brasil, na Argentina, Estados Unidos, China, Japão. Então é uma empresa bastante robusta, já tem bastante tempo no mercado, e eu trabalho especificamente na área de biocombustíveis. Então nós utilizamos enzimas para a produção de etanol, tanto de cana-de-açúcar quanto de milho. Só para dar uma geral sobre a empresa. So, I will start my presentation that was developed during my PhD at University of Campinas. The title of the project is Adaptive Laboratory Evolution of Clostridium saccharoperbutylacetonicum for Enhanced Tolerance towards Lignocellulosic Inhibitors for Butanol Production. My supervisor was Dr. Cindélia Freitas from Unicamp. As co-supervisor, I had Dr. Tiago Litabasso from University of São Paulo and Dr. Isabel Rocha from University of Minho, Portugal, in the city of Braga. Just a quick introduction. Brazil is well recognized as a big player in the bio-based economy. And here we have the sugarcane as the main substrate for bioethanol production. Just to have an idea about the size of this field, in the season 2018 and 2019, it was processed around 573 million tons of sugarcane in Brazil for biobutanol production and sugar production as well, and was generated around 143 million tons of bagasse as a byproduct of this industry. And normally, this byproduct, the bagasse and the straw, is used to generate power, to generate electricity in a cogeneration process. On the other hand, it could be used, the same material, the lignocellulosic material, could be used to produce other products with high value. So we can obtain sugars from this material in order to generate other products such as ethanol or other products. Second generation ethanol is another opportunity to use the bagasse as substrate. Just to explain, the first generation ethanol is when you crush the sugar cane and you obtain the juice that contains sugars that can be fermented by yeasts. And the second generation ethanol, we need to break down the lignocellulosic material in order to release sugars and then ferment to produce ethanol. This is the main difference, and has been described in literature that the main challenges regarding second generation ethanol is enzyme performance, is the co-fermentation of C5 and C6 sugars, the reduction of investment costs, and reduction of process time, and inhibitors. And my project decided to act in the inhibitor challenge. Normally, this material, the lignocellulosic material, needs to suffer a pre-treatment in order to release sugars. So normally we can have physical or chemical or the combination of both in order to release sugars present in the biomass. Many studies has focused on reduce the metabolic effects of toxic compounds. When I didn't explain well, but when you use these pre-treatments, normally use high temperature, high pressure, sometimes you need to use acids, and these can generate, beyond sugars, can generate inhibitors that can damage the yeasts in order to produce ethanol, for example. So the studies has been developed in order to reduce the metabolic effect of these toxic compounds. One strategy is to detoxify this material. So we can use many other strategies in order to reduce the toxic effect of these compounds. And the other strategy is to use adaptive laboratory evolution strategies in order to obtain robust strains able to tolerate these inhibitors and then ferment to produce, for example, ethanol. And my work decided to focus on the second option, adaptive evolution strategies, okay? Thinking in a biorefinery would be very important to utilize all sugars present in the biomass, the C5 and the C6 sugars. In our case, we decided to produce butanol. One is an interesting product that can be used for different applications, such as can be used in pens, in polymers, in plastic field, and can be used as biofuel. Just to have an idea about the global market of this compound, in 2012, it was produced around 3.8 million tons of butanol, and in 2018, we had an increase in about 4.6%, achieving 4.9 million tons of biobutanol. And normally, nowadays, the production of butanol can follow two routes. The most common route is petrochemical route, and the second one that is more sustainable is the biochemical route using bacteria of the genera from Clostridium species in a fermentation called ABE, because beyond produce butanol, this fermentation produce acetone and ethanol. So acetone, butanol, and ethanol in a specific proportion. And the major bottlenecks of the ABE fermentation is the high cost feedstock, especially corn, the low titer of butanol, less than 20 grams per liter, and low yield and productivity. And the specific project decided to focus in high feedstock costs. So because of that, we decided to use the hemicellulosic hydrolysate. That is a cheaper feedstock compared, for example, to corn. All right. And many studies has tried to combine the improvements in fermentation process and strain development in order to overcome the bottlenecks described before. All right. Here the goals. So the general goals of this project was obtain and characterize mutants of Clostridium species able to tolerate the menin inhibitors present in hemicellulosic hydrolysate, contributing to sugarcane biorefinery establishment. As specific goals, in the first part of the project, we decided to screen some Clostridia strains that were reported as good producers of butanol. So I will share with you the results in the first part, where we started the studying 4 strains, and then we decided to continue with one, with the biggest, with the better butanol production, to continue the work carrying out adaptive laboratory evolution. So in the first part, we start working with 4, and then we go to working only with 1 strain. In the second part, as I said, the idea was to adapt the selected strain using adaptive laboratory evolution in order to get improved mutants able to tolerate the many inhibitors present on hemicellulosic hydrolysate. And in the last part of the work, after obtaining these mutants, we decided to characterize the evolved mutants, the evolved strains, regarding fermentative performance and genomic profile in comparison to parental strain, the wild type. Okay. So in the 4th chapter, it's the part that we decided to evaluate the 4 strains. This material was published in Biomass and Bioenergy in 2019. And the paper was Towards Enhanced N-butanol Production from Sugarcane Bagasse Hemicellulosic Hydrolysate, Strain Screening, and the Effects of Sugar Concentrations and Butanol Tolerance. So we tested 4 strains: Clostridium acetobutylicum, Clostridium beijerinck, Clostridium saccharobutylicum, and Clostridium saccharoperbutylacetonicum. For the culture media preparation we used was RCM, that is a complex media for strain reactivation, and we used another mineral media component of different vitamins and amino acids and yeast extract. All right. And the experiment setup, we evaluated the 4 strains in a batch fermentation mode in a concentration of sugars around 30 grams per liter of xylose or glucose during 48 hours of cultivation in an anaerobic chamber. It's important to say that the Clostridium is an anaerobic microorganism, so we need to work in an anaerobic condition. This is the results, the cultivation in xylose and in glucose of the 4 strains. We had here the microbial growth, the butanol yield and ABA yield, the productivity, and the residual sugars in the end of fermentation. And to summarize the results, we could observe here that Clostridium saccharoperbutylacetonicum showed the best fermentative performance regarding butanol production. The yield here was 0.24 and 0.31 here, with less residual sugar here. This with 30 grams per liter of sugars. So this was the first part of the work. In the second part, continue with this specific organism, the Clostridium saccharoperbutylacetonicum. We decided to evaluate the suitable initial sugar concentration to obtain the maximum butanol titer. All right. So in this case, we used the bioreactors in a batch mode in these conditions, 30 degrees Celsius, pH 7, with vegetation of 200 RPM and during 48 hours. All right. And for this specific experiment, we decided to mix the sugars of xylose and glucose in these proportions, trying to mimic the hemicellulosic hydrolysate. All right. And we evaluate this concentration of sugars, 30, 40, 50, and 6 grams per liter. And here we can see that 50 grams per liter was the most suitable initial sugars concentration to obtain maximum butanol titer. So the titer here was 14.50 grams per liter. we can see when we increase to 6 grams per liter, the titer is the same, and we has in the end we have more residual sugars in the end of fermentation. All right, so 50 was the suitable sugar, initial sugar concentration. We carry out more fermentations to evaluate the kinetic study of the ABE fermentation. So this is the profile. Normally what we have here, in the first phase, we have the acidogenic phase, where the cells produce acids, normally acetic acid and butyric acid. And in the first phase of the grow, and what happened when the cells produce acids, the pH decrease, and achieve a minimum, a specific number, the metabolism of cells switch for solventogenic phase, where the cells normally uptake the acids and start producing solvents. All right. We decided to evaluate the butanol tolerance of cells of Clostridium saccharoperbutylacetonicum, and for this, we carry out batch fermentations in the same conditions, without agitation in this case, because we carry out in an aerobic chamber. We evaluate the different butanol concentrations, 3, 6, 12, 17, and 23 grams per liter. The butanol was added after 5 hours of cultivation, and the samples were taken over time, and we evaluate the microbial growth as a response. And for this, we evaluate the relative tolerance in comparison to a cultivation without butanol. All right. So here we have the results. We can see different concentrations of butanol evaluated, and we can see that with 3 and 6 grams per liter, the cells did not suffer about the butanol concentration present in media. But when we go to 12 grams per liter, we can see that cells start suffering because the butanol titer. So we consider that the Clostridium saccharoperbutylacetonicum is able to tolerate a maximum butanol concentration around 12 grams per liter. So between 12 and 17. So this makes sense if you remember the last result I shared with you. We were able to obtain 40.5 grams per liter of butanol. So it's around this number. All right. And then we decided to evaluate the performance of cells on fermentation on sugarcane bagasse hemicellulosic hydrolysate. So for this, we first the biomass was treated, as I said before, by hydrothermal pretreatment in these conditions. After followed by acid hydrolysis with sulfuric acid, and then after here we were able to obtain sugars present in this hemicellulosic hydrolysate. This is the composition of the sugarcane bagasse hemicellulosic hydrolysate regarding the sugars here in this first part, and regarding some inhibitors present in this part here. All right. For the fermentation, we carry out in batch mode, in batch fermentation in the bioreactor, in the same conditions described before, and samples were taken over time. All right. And we decided to add some compounds here to facilitate the fermentation of this media. All right. And this is the performance of ABE fermentation by the Clostridium saccharoperbutylacetonicum using the hemicellulosic hydrolysate as media. As we can see here, the butanol titer decreased from 0.31, 0.26 to 0.21. And we could see more residual sugars in the end of fermentation. So these results showed for us that the hemicellulose hydrolysate is really a complex media, and it represents a challenge for the cells to ferment, mainly due to the inhibitors present in this complex media. All right, and show that we need to obtain robust strains able to tolerate these inhibitors and ferment and produce butanol. So in the second part of this work that was published in this paper, the process biochemistry, we were able to obtain a robust strains using adaptive laboratory evolution strategies, able to tolerate the many inhibitors present in the hemicellulosic hydrolysate. The name of this paper was Enhancing Acetic Acid and 5-hydroxymethylfurfural Tolerance of Clostridium saccharoperbutyl acetonicum through Adaptive Laboratory Evolution. It was published last year. Here is the adaptive laboratory evolution strategy that we used in this work. We started with a wild strain, wild type. And what was the strategy? We decided to carry out repetitive batch fermentations in increasing concentrations of hemicellulosic hydrolysate. So we started with 20%, we increased to 25, 33, and 40% of hemicellulosic hydrolysate. So we carry out many repetitive batch fermentations in the same conditions, for example, in 20% of hemicellulose hydrolysate. And when we could observe an improvement in cells regarding the microbial growth, we decided to transfer to another challenge concentration of hemicellulose hydrolysate. But we need to fix a parameter that is important to say, look, we carry out many fermentations and now the cells are good to go to another concentration. And this parameter that we used here was the doubling time. The doubling time is a parameter very important when you work with cells, with microbial cells. It represent the time that cells need to double the number. For example, one cell becomes 2, 2 and 4, and so on. So is the time spent for cells to double. So we have a specific formula that we use it for this. So when we could observe a stabilization of doubling time or a decrease in doubling time, we challenge the cells for another increase in concentration of hemicellulosic hydrolysate. All right. In the end of this process, we are able to obtain an evolved population. So in this case, we do not have isolated cells, only a population that was screened in a plate, in a solid media, to obtain then the isolated to be evaluated in further experiments. All right. Here is the results, the progression of adaptive evolution of Clostridium saccharoperbutyl acetonicum at increased concentrations of hemicellulosic hydrolysate. Here we have the doubling time, here the rounds, the number of cultivations, here could be cultivations, and here the concentration of hemicellulosic hydrolysate. So we can see that in the beginning, the condition was not so challenged, so we only carry out 6 cultivations, and then we change it for the next concentration, 25%, and we could see a variation here, and then a decrease and a stabilization of the doubling time, and so on. So in the end here, we could see more variation. Is normal because we have more inhibitors present in this condition, so it's normal to have more variation. So we can see here that after this increase, we could see a reduction and then a stabilization. So here we decided to stop the strategy of evolution and then isolate mutants to be evaluated in further experiments. This is the workflow of the adaptive laboratory evolution strategy. Here we have the wild type. In the first conditions, we carry out 5 rounds. We obtain 30 generations and so on until achieve the the evolved population here with 4% of hemicellulose hydrolysate, and then we isolate 9 colons to be challenged in a conditions containing acetic acid and hydroxymethylfurfural. And I will explain why we decided to choose this, all right? So the adaptive evolution approach was carried out during approximately 130 generations, 39 rounds to obtain an evolved population called EP40, which some mutants evolved was screened in solid plate for further evaluations. We decided to evaluate the effects of inhibitors and microbial growth. So we evaluated 14 inhibitors present in hemicellulose hydrolysate, and for that, we used the factorial design, the Pleck-Burman, using the Minitab software. And we carry out in batch mode these fermentations in 30 degrees Celsius, pH 7 without agitation. This was carried out in an anaerobic chamber. And the samples were collected over time in 8, 24, and 30 hours. And the results were analyzed in Minitab software with a significance level of 95%. And the response here were the microbial growth in 34 hours, all right? This is the 40 inhibitors evaluated, is the central point, the high level, and the low level. And here are the results. We could observe that among all the inhibitors evaluated, the acetic acid and hydroxymethylfurfural were the main inhibitors that impacted significantly the microbial growth of the cells in the concentrations evaluated, all right? So because of that, we decided to challenge the evolved cells, in a media containing acetic acid and hydroxymethylfurfural. So then we decided to challenge the cells, the mutants we call here HAC2, HAC4, HAC5. From 35 isolators, we were able to obtain 9 colons mutants, and we decided to evaluate regarding the robustness to tolerate the main inhibitors. In our case, acetic acid and hydroxymethylfurfural in the concentration of 5 grams per liter of acetic acid and 0.04 grams per liter of hydroxymethylfurfural, all right? And the samples were collected over time, and the fermentation were conducted in a batch mode without inhibitors too. I will show after that we decided to carry out a fermentation with inhibitors, in this case here, with acetic acid and hydroxymethylfurfural. In another experiment, we decided to evaluate without inhibitors, and I will explain why we decided to do that. So here we can observe that the HAC2, HAC8, HAC21, and HAC25 was the only inhibitors that were able to grow in a media containing acetic acid and hydroxymethylfurfural. And more important, the wild type was not able to grow. The wild type did not grow in this condition of containing inhibitors in the media. And this was really important because this confirmed to us that the adaptive laboratory evolution was a good strategy to obtain robust strain. So this confirmed for us that we were in the right way. All right. Here is the fermentative performance of mutants in a batch cultivation in a mineral media supplemented containing inhibitors. So we can see here that HAC25 achieve a higher butanol titer around the 16.66 grams per liter. That, if you remember, is higher than 40.5 that we obtained with wild type. And in the end, we could observe sugars present in the end of fermentation, around 16%. And we could observe another pattern here, the mutants 2, 8, and 21. in the end of fermentation, we could observe more sugars present in the end of fermentation, and they produce less butanol in comparison to mutant 25, all right? So the mutant 25 showed the best fermentative performance, in our case, microbial growth, under these conditions, and consumed 84% of the supplied sugars, producing around 70 grams per liter of butanol, all right? And then we were curious about what would be the performance of the cells in a media without inhibitors. And here in this experiment here in B, we can see that even without inhibitors, the mutant 2, 8, and 21 were not able to consume efficiently the xylose present in media. On the other hand, the mutant 25 was able to consume efficiently in comparison to wild type. All right. Here in A, we can see the sugar consumption in a condition with inhibitors. That is the same like here. So we were really curious about what would be the strategy of cells to tolerate the inhibitors. So we decided to investigate, to sequence the genome of cells in order to try to understand the mechanism of cells to tolerate the inhibitors present in the media. So we did the comparative genomic of all mutants in comparison to wild type. So for this, the four mutants were sequenced using the platform, the Illumina platform. This was conducted in CTBE, that is a national laboratory in Brazil, focused on ethanol production. And the bioinformatic task was conducted in University of Minho in Portugal, under the supervision of Professor Isabel Rocha. Here, regarding the mutants 2, 8, and 21, the summary of mutations in clones obtained by adaptive laboratory evolution. I will highlight some mutations. In this case, the mutation in the gene 47240, the type is a SNP, is a deleterious mutation. And this protein, the PTS system glucose specific component, the function of this protein is to catalyze the phosphorylation of incoming sugar substrate concomitantly with their translocation across the cell membrane. And this mutation was a stop gain, all right? Another interesting mutations that we could observe is in the gene 33520. The type is a deletion, so it's a deleterious mutation in the protein that produce the sigma factors. So it's the RNA polymerase sigma factor I. The sigma factor is described to evolve in cell wall metabolism in response to heat stress. This in bacillus, but is a regulation gene. So this gene is really important and can regulate other genes. All right. So in this case, we could observe a deletion in these specific mutants. In mutant 25, we could observe a mutation, a SNP, a type SNP, like a stop gain in the gene 46930. In this gene that produces the protein sugar metabolism transcriptional repressor. The function is a repressor of the sugar utilization genes. In this case, in the arabinose metabolism transcriptor repressor. And another interesting mutation was in the gene 33510. Is the anti-sigma factor I, is a deleterious too, and the function is this gene regulates the production of sigma factor I. So the anti-sigma factor for sig I regulation through direct interaction. All right. So we could observe more mutation, but not so interesting that could explain. So based on that, the main mechanism tolerant hypothesis that we thought in this moment was that mutant 25 would have an increase in expression of sigma factor and an increase in genes related to sugar consumption, since we could observe that mutant 25 presented a mutation in the anti-sigma factor. So probably this could increase sigma factor as a response. And we could and we imagined that could observe an increase in sugar consumption, as we could see in the last experiments showed before. All right. So the activation of defense system and genes related to transport, metabolism of carbohydrates. In case of the other mutants, 2, 8, and 21, we imagine that the response that could be a down regulation of sigma factors, since we had deletion in the sigma factor, and a decrease in some genes related to sugar consumption in this case. So the hypothesis was attenuation of defense system and more efficient consumption of acetate as a catabolic repression. All right. So we decided to do more experiments, the real-time PCR, in order to discover, especially regarding the sigma factor, what would be the main effect of this. So we decided to evaluate the expression of SIGE in all, not in all mutants, but in mutants 21 and mutant 25. We can remember that mutant 2 and 8 has the same mutation as 21. So we decided only to evaluate the mutant 21. And of course, the mutant 25 that has a different pattern of mutations, all right? And for our surprise, we could observe here that all the mutants, 21 and 25, has a very small expression of SIGE when was cultivated in a specific condition. So this indicates for us that despite different mutations in SIGE, in anti-SIGE, the answer was the same, a down regulation of SIGE. So it means for us that probably a SIGE has a pivotal, important role in increased tolerance towards acetic acid and hydroxymethylfurfural in our case, all right? We evaluate other genes too, for example, ProA here. ProA is a gene that is related to biosynthesis of proline. We can see in literature that many works describe that increase the proline in cells could increase tolerance for many inhibitors. So this makes sense. We could observe an increase in this gene in mutants 21 and 25 in comparison to wild type. All right. The GRO1 is another protein that is described in literature. Is related to correct the misfolding proteins in stress conditions. So we can see here an increase in this expression of this gene in mutants in comparison to wild type. And the last gene that we decided to evaluate was the ATPD, is a TPA, is off membrane. Normally we can observe, we can see in literature that cells that has a high tolerance to acids has an increase in expression of this gene. And we can see here again that all mutants presented a higher expression of this gene in comparison to wild type. All right. So the real-time PCR showed no expression of SIGE in mutants 25 and 21. And the last experiment that we decided to do is the scanning electron microscopy to evaluate the shape of the cells. Over the adaptive laboratory evolution, we could observe an increase of cells. They become more elongated, you know, they increase the shape and the size. So it's possible to observe that mutants were much more elongated, almost twofold in length in comparison to wild types. So here in A we has the wild type, and B, C, and D, the evolved strains. So we can observe here cells more elongated here and here. So this is really interesting and makes sense, especially when we compare with other works that decided to do the same strategy of evolution in order to obtain robust strains. So as a conclusion, among the wild type strains studied, Clostridium saccharoperbutylacetonico showed the best fermentative performance for butanol production. 50 grams per liter was the most suitable initial sugar concentration to obtain maximum butanol titer and yield. The Clostridium saccharoperbutylacetonico is able to tolerate the maximum butanol concentration around 12 grams per liter. Acetic acid and hydroxymethylfurfural were the only compounds that significantly reduced these parameters in the conditions tested, so that affected the microbial growth. The adaptive laboratory evolution strategy was able to select an evolved population from which 4 cells were isolated and were challenged in a condition of media containing acetic acid and hydroxymethylfurfural. And the mutant 25 showed the best fermentative performance of in a media containing the inhibitors, producing around 17 grams per liter of butanol and consuming 84% of the provided sugars during fermentation. And the genome sequencing identifies some mutation in genes related to stress, such as sigma factors and anti-sigma factors, and genes related to sugar uptake metabolism that could be linked to the improved phenotype. The mutants 2, 8, and 21 was not able to consume efficiently the sugars, probably due to mutation in the PTS system, as possible strategy to consume acetate as tolerance strategy. And beyond that, the mutants HAC2, HAC8, and HAC21 showed no expression of sigma factor promoted by the deletion in the specific genes. And the mutant 25 showed an increase in sugar consumption, efficient due to mutation in sugar transcriptional repressor gene, and the mutation in anti-sigma factor promoted a down regulation of SIG-I, similar to the other evolved mutants. And the gene expression analysis indicated a high expression of genes related to proton pumps, ATPase synthesis, proline biosynthesis, and chaperonina, suggesting an integrated mechanism probably coordinated by repression of SIG-I in order to tolerate these inhibitors. And in the last, we could observe some morphological change in cells in the mutants over cultivation, being the mutants much more elongated, almost twofold in length in comparison to control cells. So basically is that. Carlos, I think that's on time. Thank you. Any question?
Okay, thanks, Rafael.
My pleasure.
¿Tenemos preguntas del público?
Temos uma pergunta aqui no chat, Carlos.
Ajá, sí, del profesor Gustavo Buitrago. También hay una del profesor Daniel Uribe. Tú mismo las...
Certo. Deixa eu ver aqui. Final concentration can be no comparison. Ok. Não sei se é inglês ou português, Carlos, você acha, para a resposta?
Si te sientes mejor respondiéndola en portugués, yo creo que se entiende y cualquier cosa yo la aclaro.
Tudo bem, pode ser em português, então. Ótima pergunta. A grande questão do butanol, né, hoje ele é produzido principalmente por rota petroquímica, é a questão do preço do petróleo, né? Hoje a gente tem visto o preço do petróleo disparar no mercado, né, por causa dos conflitos na Ucrânia e na Rússia. Então, isso é uma coisa que vai a favor da produção do biobutanol por rota fermentativa, né? Hoje, os principais países que produzem o biobutanol né, pela fermentação ABE é a China, no Brasil, até onde eu tenho ciência, né? Então, até onde eu trabalhei em 2020, né, 2019, 2020. Hoje eu não trabalho mais com isso desde 2019, 2020, mas a China era o principal player produzindo industrialmente o butanol. Agora, em relação à concentração e ao yield e ao rendimento, né, eu não lembro de cabeça esses números também, mas a gente tinha que, alguns trabalhos falavam na ordem de produzir acima de um rendimento, uma concentração, um título final acima de 25 gramas por litro, 30 gramas por litro. Então, é um valor bem mais alto, né? No meu trabalho aqui, a gente conseguiu atingir no máximo 16,6 gramas por litro de butanol. Então, existe um caminho muito grande ainda para poder atingir altos títulos de butanol. Uma das estratégias para você conseguir atingir títulos maiores, que é a estratégia que a Ana, né, a nossa colega colombiana, conseguiu trabalhar, era fazer a fermentação extrativa, né? Então, à medida que você vai produzindo o butanol no fermentador, você vai extraindo aquilo, né, através de vácuo, você vai extraindo os solventes, e isso vai permitindo que você consiga ter no final uma concentração mais alta de butanol, tá? Então, o butanol ele é bastante tóxico para as células. Isso foi um dos casos que foi estudado no trabalho, né? Então, a concentração que a célula conseguiu tolerar era por volta de 12 a 14 gramas por litro. Então, a gente já conseguiu, com a nossa estratégia de evolução, que ela produzisse mais butanol, né? Então, isso já foi um ganho, mas existe um caminho muito grande ainda para alcançar acima de 25, 30 gramas por litro de butanol. Acho que é isso. Tá. Em relação a aumento do tamanho das células, né? Então, na época que eu fiz a minha tese, eu procurei bastante uma explicação sobre isso, sobre por que as células aumentavam de tamanho, mas eu não consegui achar uma coisa muito consistente para explicar o porquê do aumento dessas células, né? Pode ser, essa é uma suposição minha, que pode ser para aumentar um pouco mais a superfície de contato da célula, para poder entrar mais substrato dentro da célula. Então, isso é uma hipótese que eu acho que faz sentido, mas teria que ser avaliada também, né? Mas não é muito descrito na literatura o porquê desse alongamento das células. Temos mais uma aqui.
Una pregunta de IBUN.
Sim. Essa última, né, Carlos?
Sí.
Sim. Tá. Olha, interessante essa pergunta também. Normalmente, quando a gente faz um trabalho de evolução igual eu fiz aqui, o mais interessante, assim, a cereja do bolo, né? Não sei se fala em espanhol assim, Carlos. O que seria interessante para a gente provar que a mutação que a gente obteve na evolução adaptativa foi responsável por aquela, por exemplo, aumento da tolerância ao ácido acético e ao hidroximetilfurfural. O ideal seria a gente pegar a linhagem selvagem, a wild type, e a gente fazer a mutação, né, induzir a mutação, por exemplo, a wild type, lá na mutação do sigma factor, por exemplo, que a gente descobriu, e a gente fazer o desafio. Então, normalmente, os trabalhos fazem isso, né? Eles fazem a evolução adaptativa, descobrem uma série de mutações, avaliam quais fazem mais sentido e pegam a linhagem selvagem e fazem a mutação para poder avaliar. Isso chama Reverse Metabolic Engineering, né? Que seria a engenharia metabólica reversa, tá? Então, essa é uma estratégia muito interessante. Eu gostaria de ter feito, mas eu não consegui fazer no meu doutorado pela questão do tempo, tá? Agora, se eu tivesse que escolher uma mutação para ir mais profundamente, eu escolheria com certeza a do Sigma Factor, né? Eu faria a deleção do Sigma Factor para poder fazer essa engenharia, para ver se realmente esse gene era o responsável pelo fenótipo que a gente conseguiu no final do trabalho, tá? Não sei se respondi, acredito que sim. Mais alguma pergunta, Carlos? Mais alguma?
No tenemos más preguntas aquí. Sim, mas
Tá bom, se alguém...
na minha cabeça estava pensando naquilo das múltiplas mutações que acontecem, sim, e
Uhum.
você, e tú diste mucha claridad en que aún hay, digamos que dudas, ¿cierto? Y cómo se verificaría cuál de esas mutaciones, pero seguramente hay un poquito de cada una, ¿eh? Porque pues el metabolismo está todo interrelacionado, ¿sí? El crecimiento, el alargamiento de la célula, por ejemplo, es algo que es muy interesante para tratar de explicarlo.
Sim, mas assim, interessante, Carlos, que apesar de, assim, não são muitas mutações que a gente obteve, sabe? Existem trabalhos na literatura que fazem evolução também e que obtêm milhares de mutações, né? Então, acaba ficando mais difícil você tentar explicar ou achar um padrão para você explicar o porquê a célula se tornou tolerante a, por exemplo, aos inibidores. Então, o que, que eu tentei aqui era justamente ver quais mutações estavam relacionadas dentro de um mesmo segmento, por exemplo, consumo de substrato, ou, por exemplo, no Sigma Factor ficou um pouco mais fácil, né? Porque todas as quatro linhagens, elas apresentaram mutações ou no Sigma Factor ou no anti-Sigma Factor. A princípio, isso pareceu uma coisa contraditória, porque parecia que uma ia ter uma alta expressão e as outras células teriam uma baixa expressão do sigma factor. Então a gente decidiu fazer o real time, né, o PCR em tempo real do sigma factor para a gente poder avaliar o que estava acontecendo em relação à expressão desse gene. E para nossa felicidade, nós observamos um padrão parecido, que era uma baixa expressão desse gene, o que nos fez acreditar que você fazendo uma baixa expressão, uma deleção desse gene, você poderia aumentar a tolerância dessas linhagens, no caso, para os inibidores. Mas eu concordo com você, todas as mutações, elas fazem sentido. Então, não só o sigma factor, mas a questão do consumo dos açúcares, elas estão trabalhando em conjunto para que a célula consiga suportar aquela condição, né? Então, elas evoluíram para aquilo que era como elas conseguiriam sobreviver naquela condição. Mas, assim, que nem eu te falei, se a gente fizesse a engenharia evolutiva reversa, a gente conseguiria provar qual peso teve cada uma, né? Então, se a gente, por exemplo, fizer a mutação só no sigma factor em uma linhagem e ver qual é o resultado, e pega uma linhagem selvagem e faz a mutação só no gene no repressor do metabolismo de substrato, e a gente vê qual o resultado. Então, a gente vai conseguindo avaliar de maneira parcial, assim, sabe?
Ok, sí, sí, sí. Y con certeza debe tener efectos combinados interesantes. Tem mais una pregunta, hay una pregunta más de Mariana Bustamante que creo que puede ser de la última y con eso
Sim?
concluiríamos. ¿Sí? La tienes tú ahí en el chat, ¿cierto?
Tá. Não, nós não avaliamos essa estratégia de utilizar a fração, né, de solvente, né, um método de fração de solvente para separar os compostos inibitórios. É uma possibilidade, sim, eu cheguei a ver isso na literatura, mas o foco do trabalho, desde o início do trabalho mesmo, era a gente fazer estratégias de evolução adaptativa para obter células tolerantes a esses inibidores. Então, a gente queria ter células robustas. Isso aí seria uma questão mais de melhoramento de processo, né? Então, entraria mais naquele grupo que eu falei, por exemplo, de fermentação extrativa também, né? Onde você consegue, por exemplo, extraindo butanol. A gente sabe que existe, por exemplo, resinas que você consegue também absorver os compostos inibitórios, você pode usar carvão ativado. Então, existem uma série de possibilidades, né? Então, na minha apresentação, eu falei dos métodos de detoxificação e também, né, na questão de obter células robustas. Então, a gente acabou optando pelo segundo caminho, tá? Mas, assim, é um caminho muito interessante de se explorar e provavelmente a combinação, né, das duas coisas também. Então, pode ser você, como eu falei, estratégias de processo e também melhoramento de cepas, né? Então, acho que o caminho pode ser esse.
Sí, también concuerdo que una combinación del mejoramiento con con esas estrategias para retirar los inhibidores, para retirar el solvente, pueden incrementar, como tú nos decías al principio, y tal vez llegar a esos 25, o por lo menos aproximarse a esos 25 gramos por litro. ¿Sí? Que al fin y al
Sim,
cabo, pues tenemos que pensar que esa materia prima que ha estado disponible por tanto tiempo, ¿sí? Como el petróleo, pues va agotándose y nuestra economía tiene que mudar hacia otras materias primas, hacia otros procesos.
sim.
Rafael, muchísimo obrigado. Muchísimas
De nada,
gracias por
Carlos.
tu presentación. Ana
Eu
María
posso
Set,
passar depois para você, Carlos, os artigos, se você quiser compartilhar com o pessoal, para ler com mais detalhes, sabe?
sí, por favor, envía
Tá
para
bom?
nós.
Compartilho
Gracias.
sim. Eu que agradeço o
Agradezco,
convite.
ok, agradezco a todos los asistentes, especialmente, muy especialmente a Ana María Seti, que fue la persona que hizo este contacto. Hola Ana María, un gusto en
Hola,
verte.
profesor, ¿cómo está? Un
Muy
saludo
bien,
para todos.
gracias.
Hola, Rafa.
Y con
Oi, Ana.
muchas ganas de tenerte por aquí. No sabía que trabajabas en esta parte de la fermentación o que en algún momento trabajaste en fermentación con extracción. Sí, me
Sí, con
parece
fermentación
un tema
extractiva.
muy interesante, sobre todo yo que soy ingeniero químico, pues ahí es más de nuestro quehacer.
Exactamente,
De
sí.
pronto,
La idea fue que Rafael trabajara con esa parte de evolución adaptativa. Bueno, yo también, digamos que trabajamos en conjunto y yo abarqué más la parte de proceso para retirar ese butanol que es inhibitorio para las células. Entonces también tuvimos resultados bastante interesantes que espero en un futuro próximo poder presentarles.
gracias. Bueno, ya nos pasamos del tiempo y hay mucha gente con compromisos académicos, entonces tenemos que cerrar la, la, la sesión. Muchas gracias a todos por su participación.